Welcome back to the course Fundamentals of General Immunology. We are going through Module 1, which is Immune System as a Dynamic Defense Network, and this is our lecture number 2. The title of the lecture is Immune Surveillance Across Tissues and Barrier Sites. So when we define immune surveillance, it covers three major areas. The first one is continuous monitoring. When it comes to continuous monitoring, that stands for the immune system continuously screen for the damaged tissue or the spaces which are requiring repairs, as well as any entering or any entry of the pathogens towards the host body. Then comes the primary targets. So the primary target is to detect the pathogens, damaged tissue and abnormal cellular changes. So once it is targeted, they will be destructed in the that's the third part. The ultimate goal is to destruct, eliminate the threats which are coming, which are harmful for the body, which include the tissue. Once the tissue is damaged, that they also releases a lot of chemicals which are not good for the human body, and therefore the overall immune system does the surveillance very critically in order to remove all those interrent or anything which is harmful for the body. That includes all those debris coming from the cellular degeneration and also from the entering pathogen. And in the last class we discussed that the pathogen classification broadly is defined by viruses, bacteria, fungi and parasites. So when we define the immune organs, there are two major categories to that. One is called primary immune organ and the secondary immune organ. The primary immune organs are actually lymphoid organs, and there are two major organs in that. First is bone marrow and the second is thymus. So bone marrow is actually a part of long bones, and the shaft of the long bones contains this bone marrow, and this bone marrow is the site of B cell maturation. B cell stands for B lymphocyte. And then the second organ is thymus, which is located in the anterior mediastinal area, and these are bilobed structure. It is having a small globules inside which are segregated by a septa, and one can always take a cross section of the thymus and look carefully what is there inside. And if we see a cross section of a thymus, the thymus is categorically divided into two zones. The outer zones is what we call cortex, and the middle zone is what we call medulla. So this medulla and cortex are actually the part of each and every organ system. Particularly in the immune sense, we are targeting these two areas as cortex and medulla. In the medullary region, sometimes because these areas are defined by multiple cellular processes, most of the cell types after degeneration can go there and make some kind of an area rich on those degenerated cell population. These are called as Hassel's corpuscles. Hassel's corpuscles are actually a specific area in the thymus that is located in the medullary region, and these are marked by the degenerated cell population in the thymus. Thymus is very critically the site for T cell maturation. If you see here, this is the site for T cell maturation, and the bone marrow is the site of B cell maturation. So B and T cell population names comes from bone marrow and thymus respectively. Colloquially, so people are saying this term based on the bone marrow and the thymus as a site for B cell and T cell maturation. Since these are the primary lymphoid organ, any deviation in these organs or any malfunctioning of these organ can lead to multiple disease condition. For example, if the thymus is not functioning properly, people will suffer from T cell immune specific response will be in people with having malfunction of the thymus will have less response towards the T cell population. There is a syndrome. Syndrome is always called as the collection of disease. So the syndrome is called DiGeorge syndrome, and this DiGeorge syndrome is a condition where the thymus got malfunctioned because of lack in the gene regulation for a particular chromosome. So there are specific genes which are modulating the developmental stages of the thymus. So in DiGeorge syndrome, the thymus is not functioning properly, and therefore there is a lack of T cell immune response in those individuals. The second is a nude mice. Nude mice stands for the mice which is lacking the proper T cell immune response. They lack thymus also, and in majority of the cases they lacks the hair follicles, and therefore they are called nude mice. These are widely used in the research related to cancer biology and also the immunological aspects. So these are the critical two organs which are called primary lymphoid organ, the bone marrow and the thymus. The secondary lymphoid organs consist of spleen, lymph node, and mucosa associated lymphoid tissue. So these are also called as peripheral lymphoid organ because these are secondary site of regeneration or secondary site of immune regulation. Spleen is actually located in the left side of the upper abdominal cavity, and these are very rich in the vascular supply, and therefore these organs in cross section differentiated into two distinct areas. One is called red pulp area, which is rich in blood circulatory system, and the second is called white pulp area, which is rich in lymphatic or lymphoid tissues. These spleens are also considered as graveyard of RBC. So it's a major site of destruction. Once the RBC completes its life, then the older RBC goes to the spleen where it gets degraded into its counterpart, which again utilized for regeneration of the RBCs in human body. So spleen is a very critical lymphoid organ, although it's a part of periphery system, but it does regulate a lot of important regeneration capacity in terms of immune cells. The second is called lymph node. Now lymph node is also a very important part of immune system, peripheral immune system, where the lymphatics are generated. So lymph node, if you see, these are the parts of lymph node which are located in different parts of the body. There could be a cervical lymph node which are present in the neck region, and there could be axillary lymph node which can locate into other body parts. And these small duct which travels through the lymph is called lymphatics, which carry the lymphatics into it. Now this lymph node, if you grossly look into the organ, this is also divided into outer cortex and the middle medulla. So the cortex is actually in the lymph node. The outer cortex is the site of B cell area. So these are called as follicular area. So the outer cortex is a B cell zone, while the medulla is T cell zone. When we say T cell and B cell zone, that stands for, these are the sites where the B cell and the T cell does its activity to the maximum extent. Now in lymph node there are two other aspects which we need to consider. One is called afferent vessels, where the things are coming inside the lymph node, and then there is an efferent vessels where the things are going out from the lymph node. So antigen loaded antigen presenting cells. So suppose there is a cell which already engulf a antigen. This comes through this afferent duct or vessels, and then after processing through the B cell and T cell population inside the lymph node, they are going out as an educator. Once they educate the B cell and T cell, the B and T cells, the educated one, is coming out through the afferent vessels. So we will see it in little detail later on. So, but these two are the critical lymphatic system or lymphoid organ which are considered as secondary. The minor one is mucosa associated lymphoid tissue, or in short it is called MALT, and these are specific tissue system which are located in tonsils. So here we can see that these are the tonsil area, and also in the gut. In the gut there is a very specific areas which are called Peyer's patches, which we will see little later, and these are the site for the mucosa associated lymphoid tissue. These are the area where you always encounter some of the lymphoid related cells which can control the infection if at all they are entering through the body system. So these are the two critical parts of the immune organs. One is primary, bone marrow and thymus, and the secondary, which is spleen, lymph node, and the mucosa associated lymphoid tissue. Let's try to understand some of the immune cells, and we could classify these as an immune cells classification. So we discussed in the last class that the whole immune system is divided into innate and adaptive. The innate immune system are the cells which are innate immune cells are the cells which are present right from the birth, and adaptive immune cells are the cells which are coming later in the life. So they are generated post education after entering of the foreign molecules such as pathogen, such as any kind of molecules which are foreign to human body. So when we look into the innate immune cell, these are the cells which are listed here. So the first one is neutrophil, and these neutrophils are actually cells which have a globular nucleus. So there is a multilobulated nucleus. One can easily differentiate these cells when we see in the blood smear. We can see multilobulated nucleus, and we can say that these are neutrophils. And in addition to that, these are stained by the neutral dyes. Eosinophils are actually having an acidic granules inside. So we can see that these three cell population, neutrophil, eosinophil and basophil, they have granules in the cytoplasm, and these granules are named based on their staining property with the dye. So these are neutral stain, these are acidic stain, and these are basic stain dyes. So neutrophils, eosinophil and basophils are collectively called as granulocyte because they have granules inside the cytoplasm, while B cell and T cell, which is a part of adaptive immune system, are without any granules. So they are called agranulocytes, which we'll see little ahead of time details about this adaptive immune cells. Now these cell population, neutrophil, eosinophil, basophil, are categorically utilized to do diagnosis in human beings. In earlier days, the concentration of or the amount of neutrophils in the blood population can tell that a person is whether suffering from any disease condition or not. For example, if there is a more concentration of neutrophil, one can always say that the person is suffering from a bacterial infection. While if there is an eosinophil increase, eosinophil is an indicator of a parasitic infection most of the time. It's not like a universal phenomena, but we could say that increase in the eosinophil concentration could give an indication that the person might be suffering from a parasitic infection, and therefore doctor could prescribe anti-parasitic drug in case the eosinophils are high. While basophils, the increased basophil is always related to allergy reactions. So in case of allergy, you always see that the basophil concentrations are very high. The similar modification of basophil is what we know as mast cell, that is responsible for secretion of histamines. So in basophil, there is a change in the processes which are happening inside the cell, and the basophil is later on converted into a mast cell, which is actually an active form of basophil that secrete histamines. And then we have three other cells called monocyte, dendritic cell and natural killer cell. These are always present as a part of innate immune system. So when we talk about innate immune system, we have to understand that these all cell population are invariably present in the human body even without exposure of any entering pathogens. While adaptive immune system, very specifically these two, A granulocyte, which we discussed without any granule, B lymphocyte and the T lymphocyte, are actually part of adaptive immune system, which are generated later on the life, later on the exposure of the antigen by getting the response from the antigen presenting cell, educating the immune system how to get rid of those pathogens. So these are very specific cell population. And then we have other immune cells, some of them I already discussed, mast cell, and these are the plasma cell, which is actually a memory cells. Suppose if there is a body system, and if there is an antigen entering inside the body, the first line is innate immune system. When it fails, then adaptive comes to the picture, and at most of the time this adaptive will clear the pathogen from the system. And the part of information of the adaptive system are stored in the form of memory cell, which is otherwise called as plasma cells. So these are also very critical cell population that is responsible for memorizing the antigen in future exposures. So these are some of the classification of the immune cells. So till now what we have understood that there are two distinct parts of the immune organs, immune systems. One is called primary lymphoid organs and the secondary lymphoid organs. Then we have immune cells, which are either divided into innate, adaptive and other cell types. So we will move further and see that what are the things which are happening when the antigens or any foreign molecules are entering inside the body. So once the pathogen or anything which are not self, we simply say it's a non-self, which are coming from outside, entering inside the body system, there are defense mechanisms. The primary defense, chemical defense and biological defense, which are listed here. So once the pathogen is coming into the system, these are the mucus layer actually. These are the thick mucus layer which are secreted from this epithelial layer. So this mucus can trap those entering pathogen or antigen so as to not allow them to cause the effect. And these mucus is secreted by this layer of cells, and these layers of cells are very tightly bound to each other so that the antigen may not percolate to the bottom. That means there are cells which can secrete this mucus to trap the antigen, and same time these mucus are not allowing anything to go down so as to reach to the body cavity to do the problems. And therefore these cells are very tightly bound to each other. There are so these two cells are joined by various junctions, and there are few which are listed here. One is called tight junctions and the other is called desmosomes. So the tight junction is actually a fixed junction which will allow very tight adherence in between the two cell populations so that nothing can go leak out from the system. And the desmosomes is actually a part of filament, intermediate filaments, which are helping the two cell population to communicate with each other. So these two, apart from that, there are gap junction as well, and this gap junction could be a junction which will allow the passage of the solute or ions very critically into the cellular system. So these junctions are very important, and therefore I listed here to give an introduction to the junctions as well. So these are kind of a physical defense which are present right in the skin layer. After that, there are a lot of things which are secreted by the cellular system. Some of them are lysozymes, which are actually lytic chemicals. Once they will touch into this or touch the pathogen coming from outside, they will immediately lyse it out or try to lyse it out. The second is acidic pH, which is harmful for anything. So the more amount of H plus ion into this mucus will allow that the pathogen to degrade instantaneously. And then we have lot of antimicrobial peptides, and these peptides are a small stretch of amino acid stretch, and these amino acid chains, small chains of amino acids, are actually perturbing the cell wall of bacteria in order to destroy them critically right there in the entry point. These are very well studied in research aspect. Some of the antimicrobial peptides are example I could give a defensin, which is actually a very strong antimicrobial peptide. So these three chemical defense which are there in the system can destroy the entering pathogen right there. And the third is the biological defense. So we can see that the commensal microbiota, so these are the pathogens which are entering into the body system. And then we have our useful bacteria which is there in the gut wall. They won't allow them to go inside. So they are kind of a shield which are happening into the body cavity, and because of that reason they are not able to penetrate into the body system freely, or rather to say that they will compete with each other so that they won't allow them to go inside, and therefore protecting in a different way altogether. So these are the first line of defense where the physical, chemical and biological systems comes into picture and protect the body cavity in order to avoid the pathogen entry into the system. Right. And then we will look into the localized surveillance, and we will just study three system here: skin, the lung epithelial and the gastrointestinal tract, because these are the major site where the pathogens and the antigens are interacting. So if we see skin is an outer covering of the human being. As we say that these are the largest organ of a human body. The outer part is called epidermis, which is the outer layer, and the bottom layer is called dermis, and just below that we will find the muscle layer. So these are all muscle area which is there in the lower side of the dermis. Now this epidermis is made up of multiple layer of the cells, and these are tightly bound with those all junctions which we discussed, either gap junction, desmosomes, gap junction. There is another junction also which is called adherent junction, but this is beyond the context of this discussion as of now. But we critically look into that this epidermis area contains lot of cells which are very important, and these cells are actually a phagocytic cell, so they can trap the antigen and digest them right away after getting contact with those antigens. So these cells, some of the cells are called phagocytic cells of the skin, and these are called named as Langerhans cells. And Langerhans cells are having a lot of this elongated structure, which we call pseudopodia-like structure. They have a capacity to capture the antigen and then engulf it out, and then further to process and present it so that the antigen presenting cell can identify that these are the antigens which are not self for the body system, and then accordingly respond to the pathogen destruction. So these are the upper area of the epidermis. The second is dermis, and dermis is full of blood vessels and also some of the T cells which are present, some of the macrophages. These are the big cell which they can also eat pathogen, therefore they are called phage or phage. These are eater. And then we have mast cell, which is actually a cell which can produce histamine at the time of allergic reactions. So these are the cell population which are present in the dermis. So the skin is also a resident of multiple cellular type in order to protect the body system from any entering pathogens. Second is respiratory tract, and respiratory tract, as we know, that it's very important system. So because they can directly access through the nostril and the airway epithelium. So the person who getting infection is most of the time is through the respiratory route, while inhalation of the aerosol borne pathogens. So most of the pathogens which are airborne are mostly coming through the system of respiratory tract and entering inside the body. So we need to understand what are the in-depth mechanisms which can help the respiratory tract to build up in a proper way. So the first one is the airway epithelium, and if you critically look into the airway epithelium, this epithelium are having a cell and a ciliated structure. This cilia is very important because they can beat in a direction so as to expel out the pathogen in a proper way. So these, and also they secrete huge amount of mucus, and this mucus can trap these pathogens which are entering from the airway epithelium or through the airway system. So this mucus can trap the pathogen in a very effective way, and then accordingly try to destroy based on the acidic pH or based on the specific amount of enzymes such as lysozyme present there, or else they can be phagocytized by the cells which are present in the airway epithelium and then pass on to the next level so as to raise the adaptive immune response. So these are very important area and the first line where the entering pathogen can come into contact. And if at all the pathogen pass through all these things, they can reach to this alveolar space which is in the bottom of the lung, and that's the actual site where this alveolar epithelium can help to exchange the gases, the CO2 and O2. In addition to that, we have this small secretory immunoglobulins, which we will see little later, that what are these immunoglobulins, why it is called secretory immunoglobulins, and these are also present in multiple other areas. So for time being, we will just say that these are called secretory IgA. This is a class of immunoglobulin. One such class of immunoglobulin, this is defined as immunoglobulin type A, and since these are secreted in nature, these are liquid in nature, therefore they are just secreted out into the system, and therefore these are called secretory immunoglobulin A. Apart from that, we have this big cells, same like the Langerhans cells which we see in the skin layer. We have also alveolar macrophages that can engulf this pathogen and then process it out for body defense system. Then we have this specific epithelium, which I already explained, that helps in the gaseous exchange, and also they provide an environment of flexibility to the system, so that more and more flexibility can be given to the airway epithelium. So these are the deep level epithelium. Just to mention here that these airway epithelium expresses lot of proteins on their surface. So these proteins are either expressed on the surface or will be nicely expressed on the cellular architect, so as to give access to the other molecules. In addition to that, this receptor, and one such example is the sialic acid receptor. So the sialic acid receptor are very critical receptor which are present in airway epithelium or in the alveolar system, that give attachment to the influenza viruses, and therefore or coronaviruses. So these respiratory related viruses can attach to these receptors which are expressed in the alveolar epithelium, and these processes of attachment to the sialic acid receptor and the pathogen is very critical, and people are always trying to understand how we can abort this binding of pathogen to the sialic acid receptor. So these particular protein which are present in the alveolar epithelium is very important. And then finally to the gastrointestinal tract, and these are the gut area where we always, so inhalation is one route where we take the air that goes through the lung, and the second entry point is through the food. So if the food is contaminated and the food carries some of bacteria, some of the pathogens, then obviously it goes to the gut. Most of the time the acidic pH of the stomach can degenerate those specific pathogen, but if at all they bypass that acidic pH, they can go to the gut lumen. So these are the luminary area. And if you critically see, these are again the secretory immunoglobulin A which are present in the gut area, and then we have this epithelial layer which is nicely decorated with this ciliated epithelium, which is actually beating in a direction so as to give fluidity to the system. Mucus is also secreted here. One critical point to remember here, that these beating of cilia and the secretion of mucus is very critical here, because if there is a deviation in the gene expression of this cell, then we can see that the mucus secretion is also stopped or reduced, which can indirectly affect the multiple pathogen entry into the host cell. So these are the areas which are epithelial. Out of all this epithelial area, we can see here these are very specialized cells which are called M cell, which are otherwise called as microfold cell. These microfold cells are also called as M cell, are the cell, as you see that the name itself indicates microfold. Micro stands for small. So instead of bigger folds which are present in other epithelial area, these folds are smaller or absent in this cell. So M folds are very critically the cells of the intestinal area, which helps to trap the pathogen and simply pass on to the next level. So these are actually a passager. So they can simply pass on the pathogen from outer area to the inner side, and this inner side is actually an area which we have studied when we discussed about the secondary lymphoid organ called Peyer's patches. These Peyer's patches are very rich area where we can find out macrophages, dendritic cells, and also T cell population. And therefore, once these M cells can capture the pathogen, they can directly enter inside the Peyer's patches, where this macrophages can engulf this pathogen, process it out, decorate it itself into the surface so that the immune cells can understand this. For example, the T cell can recognize this antigen as a foreign and try to destroy itself right there. And therefore, these Peyer's patches are important, and these M cells are very critical because they can give passage of pathogen towards the site next to the level, which will allow the destruction of the pathogen there. So the GI tract is also very important site where we can see lot of immune sentinel cells are present. So in total, what we have understood till now, that we have primary and secondary lymphoid organs. We have different kinds of cell, either in the innate immune side, adaptive side, and there are some specialized cells such as plasma and mast cell. And then we have 3 layers of the cells which are tackling the entering pathogen. For example, the skin layer is having some cellular types such as Langerhans. Then we have a lung epithelial where you have another alveolar macrophages. And finally, in the gastrointestinal tract, we have cells such as M cell, which is helping to catch the pathogen and pass on to the next level so that the adaptive immune response can be generated. So these are very critically synchronized processes which are happening into the system. So looking into the loops and trying to link all which we have discussed till now. So what's happening is basically when there is a pathogen entry, the pathogen is engulfed by these antigen presenting cell or macrophages. And these macrophages, or we can also say these are dendritic cells, because these are having a dendrites which are coming out from the surface to help capturing the pathogen. These are pass on, slowly migrate and pass on to the next generation, and this travels to the nearest lymph node. And this lymph node, as I told, the outer area is actually a B cell area, the follicle area, and the middle area is what we call the T cell area. So this dendritic cell which are carrying the pathogen comes to the site and show this pathogen part to B and T cell population. The B cell and T cell, after recognizing this pathogen as a foreign, can sensitize or can activate themselves in a way so that they can go back to the site and destroy the entering pathogen right there itself. So this whole cascade is very synchronized. So the part is starting from peripheral tissue, the entering point, then we have a lymphatics which are involved there, and finally the effective response where the T cell goes and destroy the pathogen into the site of entry. So these all synchronized process is very critically controlled from the tissue level, also in the gene regulation level, and the immune system is highly orchestrated when it comes to the response towards the foreign molecules. Sometime, as we discussed in the last class also, the regulation of this immune system is critically important in human body, and if we fail to control this immune regulation, sometime we may end up in getting the extra immune response which is toxic to the human body. So we don't want an extra immune response. Instead, we want a controlled immune response. So we should have a balance of immune response, not to be very high, not to be very low. So it should be a very balanced immune response. And finally, to summarize, that we have a skin, lung, gut, we also have multiple other organs, but we summarized here in a critical way that these 3 are the actual site where most of the things comes. Like skin is the site where things are coming. Lungs are the site because we inhale a lot of things every day, most of the time. Gut is the site because we take a lot of food which are either containing the pathogen or any kind of harmful substances, and it has to be processed through the gut epithelium. So these 3 are the important. And then we have the counter mechanism where the things can curtail in a different way after sensitizing the immune response in a proper fashion. Thank you.